Hello, everyone, and welcome to today's webinar, Biomarker Breakdown, Combating Global Fungal Virulence, my apologies, Diagnostics and Resistance with Dr. Jennifer Geddes-McAlister. I am Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I would now like to welcome our speaker, Dr. Jennifer Geddes-McAlister, Associate Professor at University of Guelph. For a complete biography on Dr. Geddes-McAlister, please visit the Biography tab at the left of your screen. Following our presentation today, Dr. Geddes-McAlister will have a brief conversation with Stephanie Samra, Senior Global Market Development Manager for Translational Research at Thermo Fisher Scientific. Immediately following, we will open the floor for live questions. Dr. Geddes-McAlister, welcome. Thank you for joining us. You may now begin your presentation.
Hi, I'm Dr. Jennifer Geddes-McAlister from the University of Guelph, and it is a pleasure to speak to you today about some of our research, in particular our biomarker breakdown and how we're using proteomics to combat global fungal virulence, diagnostics, and resistance. So the global impact of fungal disease is quite staggering. There's over 3.8 million deaths per year that are reported and associated with invasive fungal infections, and mortality rates range up to 90% depending on the pathogen, the location, and the availability of antifungal drugs. Some of the other challenges that we face include inadequate surveillance and diagnostic programs, where we have difficulties monitoring baseline levels or population levels of fungal pathogens and fungal infections, as well as a distinction between bacterial and fungal disease, however, not necessarily information on antifungal resistance profiles to diagnose and effectively treat the infections. We're also hampered by increased geographical spreading associated with international travel, where we have fungal pathogens going to new regions of the world and adapting under climate changes, which promote these pathogens to emerge as new infectious agents, or even reemergence of some that we haven't seen in many years. And then lastly, one of our biggest challenges is the limited antifungal agents that we have currently available. One of the biggest challenges for antifungal agents is homology between the target within the fungal pathogen and within human host. And oftentimes with antifungal drugs, we see a high cytotoxicity towards the host. We also have increasing rates of antifungal resistance. So very few drugs that are effective against these fungal pathogens, and we're using them quite a bit, or we use them in our initial mainstay drugs to treat infections, leading to resistance over time. In my lab, one of our approaches to really tackle fungal diseases, we take a One Health approach. And the term One Health really looks at the interconnectivity across animal health, human health, and the environment, and how they all relate together. And particularly in my lab, we're interested in studying fungal pathogens and how they interact with the immune cells and with the host. We use mammalian systems to study these pathogens, but we also look at how they interact with bacterial pathogens as well within a co-infection setting, for example, and that interplay across the different systems. We then use some of this information to identify novel antifungal agents and potential targets for antifungals, and explore that in a medical context towards human health and identification of new antifungal drugs that we can test for efficacy. And then from the environmental perspective, we look and see the production of some of these drugs or biologics within a plant-based system, as well as fungal disease within an agricultural context and how that impacts the environment. And one of the particular examples that my lab is interested in really does explore that relationship between fungal disease in an agricultural context and fungal disease within a clinical context. And so this is an example of a One Health problem and how we have a connection driving antifungal resistance. For example, half my lab studies Fusarium graminearum, which is the fungal disease of wheat and cereal crops, wheat, barley, corn, and really looking at how this pathogen infects the cereal crops and reduces grain quality and yield, but also produces mycotoxins that are harmful to animals and humans upon consumption of contaminated grains. And so that in itself presents a One Health challenge in the sense that you have this environmental pathogen impacting animal and human health. But in a larger context, if we look at this where we have prevention of fungal infection within the agricultural setting, within the field, and that the first stage there is this fungicide application each year. And typically the fungicides that are applied are azoles, or within that classification of antifungal agents. And so these azoles, or these fungicides, are applied to prevent infection in the field, but the fungicides will remain within the environment, within the soil and water runoff. And you have environmental microbes, such as Cryptococcus neoformans, which is a human fungal pathogen, that resides within the environment. And so you have this opportunity for an environmental microbe to then be exposed to this fungicide. And the context of that and where the One Health piece comes in is that if these pathogens evolve resistance and they're inhaled and produce infection within a human, our ability to treat the infections are quite limited when in the presence of resistance. We have few antifungals to begin with, and now we've actually taken one away because the pathogen has evolved to be resistant from exposure within the agricultural context or in the field. And so this One Health problem of really looking at prevention of fungal disease within the field and exposure of microbes to these fungicides and their evolution towards resistance is a tangible example of a One Health problem that we see, particularly with fungal pathogens and fungal disease, and how we're trying to tackle that to increase global health or improve global health. And so my lab has defined 4 pillars to tackle fungal disease. And this is our specific approach for doing the work. The first pillar being prevention. And here, 3 of my graduate students, Davier, Jacob, and Oscar, are really looking at anti-virulence strategies to disarm the pathogen and promote clearance of the pathogen by the host immune system. Pillar 2 is diagnostics, and a previous graduate student in my group, Ben Muselius, and an undergraduate student, Lauren, are studying diagnostics and organ atlas, and how can we really define the proteins that change within the host and within the pathogen throughout stages of infection, but also within different temporal or spatial regions too. So how does the pathogen adapt within the host? Pillar 3 encompasses monitoring, where we're really quite interested in looking at host-pathogen interactions. So how does the pathogen evade the host immune response, and how does the host defend itself from infection? And then lastly, Pillar 4 is looking at treatment options. Can we develop new antifungal agents to be effective? And more importantly, if we have that evolution of antifungal resistance or the occurrence of antifungal resistance, how can we tackle that and overcome or reverse resistance within these pathogens? And so using these 4 pillars then is how we really build our research program to tackle fungal disease, and we use proteomics to do this. So the pathogen that I'll focus on today for our talk is Cryptococcus neoformans, and this leads to cryptococcosis. So the fungal pathogen itself is typically, it's ubiquitous within the environment, within eucalyptus trees and pigeon guano, decaying tree barks and soil. And upon disruption of those environments, the fungal cells can be inhaled into the lungs of a human host. Now, within a healthy individual or immunocompetent individual, the immune system will become activated. Macrophages will work to phagocytose the pathogen and clear it, and your T cells will be activated to kill the fungal cells as well. However, critically, within immunocompromised individuals, such as individuals with HIV/AIDS, co-infections, as well as patients receiving immunotherapy, the immune system is not as strong to combat the fungal pathogen. So you may not have full phagocytosis by macrophage, or you may not have activation of the immune system by T cells. And that interplay then allows the pathogen to actually reside within the host for indeterminate amounts of time. And it can also disseminate. And so that dissemination is where we see the largest impact of infection, where the pathogen can cross the blood-brain barrier and enter into the central nervous system. And during this phase then can cause cryptococcal meningitis. And that's where those high rates of fatality come into play. And so to cause this and also considerations for disease, we can look at global distribution. The majority of cases of cryptococcosis occur within sub-Saharan Africa because of that connection with HIV/AIDS patients. But with the increase and availability and effectiveness of antiretroviral therapies, the numbers of infections have decreased over the years, which is excellent. However, we're seeing increases in areas that we did not experience cryptococcal infection before, such as the Pacific Northwest. Vancouver Island has also had an outbreak of cryptococcal species. And to cause infection, the fungus itself produces different virulence factors. And one of these in the image in the middle here is this polysaccharide capsule that surrounds the cells. And it's kind of this halo that forms itself around the fungal cells, which prevent macrophage from phagocytosing the pathogen itself, or it also protects it once it's in the macrophage. The pathogen also produces different virulence factors, melanin being one for protection against reactive oxygen species, extracellular enzymes that assist with crossing the blood-brain barrier, and thermotolerance, allowing it to adapt from an environmental temperature into a human physiological temperature. And it really is that power play or that point of an interaction with macrophage that drive the immune response towards fungal clearance. We do have antifungal agents that are available to treat cryptococcal infection. There's 3 classes that are primarily used. One of them is highly cytotoxic and neurotoxic and is delivered only under hospital administration. And our other mainstay one is fluconazole, which is an azole-based drug that patients can have to undergo therapy for over a year or even a lifetime, depending on the infection. And these azoles are the ones I referred to in that One Health context of connections between the field and the clinic. And as I mentioned, we use mass spectrometry-based proteomics, as well as advanced bioinformatics tools and workflows to really study these biological systems. A lot of our work is based on dual perspective profiling, where we're looking at the host response to infection and how the host is adapting, but it's also equally important to us to understand how the pathogen is adapting to those different environments and what proteins are changing within the pathogen that may represent new virulence factors, new druggable targets. And so as a generic workflow here, we begin with different biological samples. These may include cell pellets, secretome might be a fluid or a tissue as well. And we digest the proteins into peptides. We use primarily a bottom-up based approach. And those peptides are then measured on a mass spectrometer. We then map the peptides back to the proteins using different computational platforms. And we then use this to get information about which proteins are being produced within the biological systems and to what abundance. And are there changes depending on the different conditions, infected versus uninfected, as well as looking at deletion strains and how that impacts the proteome. And so there's many different applications that we apply proteomics to really understand and tackle those 4 pillars against fungal disease. And so the first pillar is looking at natural sources of antifungal compounds. And this is quite a fun pillar to start with because we are using mollusks and snails from the environment to look for novel antifungal agents. And the reasons behind this are that snakes, like being sessile invertebrates, they need to have effective chemical and innate defenses to defend themselves from the environment and threats within the environment. Likewise, within Ontario, where the samples are collected, there is a lack of evolutionary adaptation to Cryptococcus neoformans. The pathogen does not typically reside within that area. It's more in temperate climates. And so the snails and mollusks have not evolved to protect themselves necessarily from Cryptococcus, allowing us to identify potential new agents that haven't seen the fungus before. And then also limited host cytotoxicity. So when we're looking at this the snails themselves and the different inhibitors and proteins that they produce to protect themselves, we're also potentially looking at natural compounds that are not as cytotoxic to a mammalian system. So it gives us a good jumping off point to really start to study what is present within these systems. And so Davier, a graduate student in my group, he went and collected samples from the environment, then developed an extraction protocol and extracted proteins and peptides, crude and clarified extract, to identify if they do have these antifungal properties. And what was really interesting is that, and I will admit, I was quite skeptical at whether this would work or not in the beginning. What was interesting is that the different extracts had different antifungal properties. So in the plot here, we can see that we have our wild type untreated cell, and we have that characteristic polysaccharide capsule that surrounds the cells. But if we treat the fungal cells with the extract, that capsule is no longer there. So it's possible that we're inhibiting production of the capsule or even disrupting attachment of the capsule to the cell surface. We also explore different virulence factors like thermal tolerance, and in the presence of the extract, we see a significant reduction in fungal growth, suggesting that it's impacting heat shock proteins, or the ability of the fungus to adapt to that higher temperature. So Davier developed a fingerprint then for the different extracts and looked at how they impacted, like which virulence factors and to what extent they inhibited the virulence factors. And to really start to kind of pare that down and identify a specific compound or a specific target that had these effects, he then took the extract and fractionated it by size exclusion chromatography, tested out the extracts for a specific property. So in this case here, he was looking for that anti-capsule activity, and identified a fraction that had a significant reduction that led to a significant reduction in capsule production. We then performed proteomics on that extract to identify which proteins or peptides are present within the sample. And the pie chart here just is a visual representation of the diversity of proteins based on their gene ontology terms that were identified within that one particular fraction. And so what we wanted to do with this data then is we want to identify, we know that if we have the extract and we treat the fungal cells with it, that the capsule is removed or the capsule's not there, but we don't know how exactly that's occurring. And so we developed a list of proteases that the fungi produces that are associated with these virulence factors. For example, a serine protease is associated with capsule production. And so we have the extract fraction that has anticapsular activity, and we can identify proteins that are within that fraction. We then identify potential inhibitors of the serine protease. And we took this then to complement that experimental workflow and developed an in vitro or an in silico, computational workflow. So Jacob in the lab, and here's an example, this is the workflow that we've developed, would take our target protease from the fungi and using AlphaFold predict the structure of that protease. We then take examples of potential inhibitors, peptides, primarily peptides and proteins, and identify interactions based on a test data set that we have of known and positive inhibitors that interact proteases within fungal pathogens. There's not many examples in Cryptococcus neoformans, and so we expand our search to look at other fungal pathogens such as Candida, or even working within model systems like Saccharomyces. And so we take these proteases within the same class that we're interested in, and we dock our peptides or proteins that have shown to have inhibitory activity. And that allows us to then classify potential inhibitors as positively inhibiting our target or negative interactors. And so from the data set here, we can have our predicted structure of our protease that the fungi produces, and then we dock to it a peptide with potential inhibitory activity. And we then synthesize those potential inhibitors and test them out in the lab. And we start by testing on commercial enzymes, and then we proceed to testing them on the cryptococcal enzymes themselves to look for that inhibitory activity. And so the plot here then shows if we have our control treated and the production of capsule, and if we add one of our inhibitors, we see a significant reduction in capsule production. And this gives us that first glimpse into the proof of principle that, we have a crude extract from a snail. We know that it impacts or reduces the polysaccharide capsule in the fungus. We're not exactly sure how. So we use proteomics to identify what those potential inhibitors can be. We combine that with our computational workflow, synthesize the inhibitors, and then test them out in the lab against those fungal proteases. And that allows us to then replicate some of these phenotypic observations. So that in silico prioritization of the virulence regulating proteases really helped us to develop the pipeline, and then really work with structural prediction and interactor modeling to identify these and classify these potential inhibitors. And so our next steps then are to test out inhibitors against the cryptococcal enzymes and look to see their function that they have. And so that brings us over to pillar 2. Pillar 1 is really focused on the anti-virulence strategies and, tackling the pathogen. How can we disarm the pathogen? How can we get rid of that capsule and promote clearance by the immune system? And now within pillar 2, we move into infection has occurred. And here we're looking at an organ atlas of cryptococcal infection with the goal of improving diagnostic strategies. We do this from a dual perspective, so the host and the pathogen profiling to understand how each of them is responding to infection. We do this across a temporal and spatial scale to get resolution from both axes, where we really want to see the initial innate immune response about 3 days into infection within a mouse model, that crosstalk point between the innate and the adaptive immune system at 10 days post-inoculation, and then at the terminal point, really where the adaptive immune system is taking over, how does that change across time? Spatially, we performed this analysis across 13 different organs and fluids from the mouse model to identify how the pathogen changes depending on the location within the host, and also how the host changes depending on the organ that you're looking at. We then built the C Proteo Atlas, which is publicly available, to make our data accessible. This will be primarily of interest to other fungal disease researchers, but you get both perspectives, and so you can understand more how the host is really working to defend itself from infection. So this is an example of the C Proteo Atlas and the different components that we've included, and we will be releasing this later this year to explore the proteins that are produced across the different organs and fluids, and also looking at their abundance changes within an organ and across time or across the different systems, and those that are common and those that are unique. And from this data set, then, we really work to discover these multi-panel or multi-protein signature panels. And in the top, the T-SNE plot here, we have our, this is the host perspective, and we've really condensed the variables within our analysis here to look at, how does the host respond during infection versus an uninfected control? How does it respond over time? Are there differences across the organs? And one of the examples we see here is the spleen across time points does not have a huge distinction in its proteome profile, but we do see a distinction between infected and uninfected. Whereas if we look at the spinal cord, there we see time also has a big factor and plays a big part, which we would expect because the fungus will infect the host in the lungs and then disseminate and cross the blood-brain barrier to enter into the central nervous system, where we may then see the spinal cord and changes the protein level there. On the flip side of this, if we look at the pathogen in the upset plot, this gives us an example and just highlights that we have these common proteins that are produced by the pathogen, regardless of the location within the host. But we can also identify, organ-specific proteins. So in the kidney, there are specific fungal proteins that we detect versus in the eye or within the blood. And this gives us insight into how the pathogen is really adapting to its environment. The heat maps here allow us to then look at rates of change of proteins being produced. So in the first column, we're looking at protein abundance changes from that 3 to 10 days. and the second column is looking at 10 to 21 days. And these particular examples from the bladder, we see that the host proteome really increases in abundance early on and then decreases over time. And the pathogen actually shows quite a similar profile for the majority of proteins that we detect, which was interesting for us to see. And so we've done this profiling across the 13 different organs and fluids. But what's quite valuable from this look in particular is that if we take the organ and we extract, like we grind it up and we plate some of the tissue, and then we do fungal load counts at the different time points, we don't detect the pathogen from the bladder, for example, until the 21-day mark, which means that you're only looking at like a chronic infection, and that fungal load has to be very high for us to detect it within this organ. Conversely, though, at the protein level, we see remodeling at the 3-day time point right away when we start looking at it. So it gives us more power for diagnostics and sensitivity with that early detection. And if we take it one step further, then we can look at these signature patterns and develop these panels from both the host side and the pathogen side. So for example, here, this is the complement C3 that is produced by the host, an adaptive immune associated protein that increases over the time course of infection, which is a great control for us to see within our analysis. And using a Pearson correlation based on the slope of this protein abundance profile, we then map other proteins that show a similar profile with that increase over time. And from these other candidates, we have stress response proteins, host proteins involved in transport, but more importantly, it gives us sensitivity that we haven't seen before within diagnostic panels. So not only could we use this both from the host perspective and the pathogen perspective, but we can actually also then begin to say it's a fungal infection, it's a cryptococcal infection, and we could even move on to resistance and looking at that as well. And so this proteome remodeling is really tissue and time dependent, is the information that we get from this organ atlas. And that if we can develop these diagnostic protein signatures from both the host and pathogen perspectives, it gives us more power for identifying the pathogen and kind of naming that infectious agent prior to culturing and being able to do that. We can also define site-specific virulence factors, and those can be putative novel druggable targets that we can then explore further on to limit the dissemination of the pathogen within the host, for example. So if we move from that pillar 1, the prevention, the antivirulence, we move into pillar 2, where we're looking at diagnostics. Now we're in pillar 3, which is really looking at the relationship between the host pathogen interactions. So how does the host defend itself from infection, and how does the pathogen evade those immune responses? We want to identify infection-associated fungal proteins, then generate and characterize the deletion strains, and assess the role of that protein in fungal virulence using an in vivo model. In the top plot here, this is the fungal proteome. And what we've done is we've plotted the proteins, their changes in abundance, whether they're grown in vitro in culture media within the lab, and then in the presence of macrophage. And what we're interested in are these proteins in blue here, and they show a significant increase in abundance in the presence of macrophage, suggesting that they're infection associated. Along the bottom, we have our infinity proteins, and those are fungal proteins that were actually only produced in the presence of macrophage, not within the in vitro media. So these infection-relevant or infection-associated proteins give us kind of a jumping-off point to then study whether they're involved in virulence and what their role is within the host system. So a graduate student in my group, Brianna Ball, she generated deletion strains. So we had 53 candidates in the beginning, protein candidates. She prioritized them into a group of 17 fungal proteins to look at further. These were based on known roles in virulence, predicted secretion or known secretion, as well as uncharacterized proteins that we could study further. And so she generated deletion strains for the different candidates and then assessed their their phenomes, looking at those virulence factors. So capsule production, melanin, thermotolerance, ability to infect macrophage, and she generated this phenome heat map. And what we observed from this was specific trends in the fungal proteins. And that's where these three categories come from. So from the trends, she was able to define a general category of antifungal properties. And this would be a gene deletion strain that shows a reduction in all aspects of fungal pathogenesis. So we're looking at decrease in capsule, decrease in melanin, a decrease in thermotolerance. The second category is ideal antivirulence. And this is where we really see we're focusing only on those critical virulence factors, but not targeting fungal growth. We don't want to push evolution of resistance to the particular drug. And the last category is infection relevant, where we look at a reduction in virulence factors, and growth is one of the areas that we can explore. And so the candidates were then mapped against these three categories. And she prioritized one protein from each of those categories, or one corresponding gene, deletion strain, and assessed it within an in vivo model. And that's the data that's shown here. And what we saw with this particular candidate is that deletion of this gene led to a complete reduction in fungal burden within the mouse model, as well as survival of the mice to the endpoint of the trial compared to the wild type strain. And this gave us insight into the fact that this candidate, this protein that we defined as being infection relevant, really does have a role in virulence within this in vivo model and is a potential druggable target. And not only an antifungal target, but also it could also prevent infection. And so I have an example here of how we took one of these targets. We used proteomics to explore the difference between the wild type strain and the gene deletion strain to give us some ideas of what it was involved in. This particular target was associated with extracellular vesicle composition. And we then set up a vaccine trial with mice where we did vaccination with extracellular vesicles derived from wild type fungal cells versus from this deletion strain fungal cells, and how the immune system actually changed. And so in the blot here, we see a huge increase in the immune response in the presence of these extracellular vesicles from a deletion strain. So this is just one example of the ones that we've explored where we start to connect the protein profiling from the beginning, and that really looking at that infection-associated proteins, and then moving that through entire phenotype profiling and in vivo models to potential new therapeutic agents. And it allows us to characterize the role of these proteins in fungal virulence as well as dissemination, and to propose and evaluate some of these new therapeutic interventions, such as vaccines. And now what we're doing is combining this information with computational and high throughput screening platforms to identify inhibitors or compounds that will target these specific proteins that we're interested in. And so that then takes us across to pillar 4. So we've looked at our prevention, we've looked at diagnostics, we've looked at the relationship during infection, and now we want to be able to treat infection. And one of the biggest challenges is overcoming antifungal resistance. And so how can we do that? And we tackled this by starting with evolving resistance within our lab-associated strain towards fluconazole, which is one of the mainstay antifungal agents. So we took our wild type susceptible strain, and over about 2 months of exposure to fluconazole, evolved it to be resistant. We then looked at changes in the proteome between the susceptible strain and the resistant strain. We also added in a clinical isolate to look at common changes that occur within resistant strains. And that led us to investigate a specific target. In the plot here, we see we have our wild type, which is susceptible, and our lab evolved, which is resistant to fluconazole. In the absence of a drug, both of the strains go well. In the presence of fluconazole, we see that the wild type is susceptible to the drug and unable to grow, but our resistant strain still can. Then using proteomics, we investigated what proteins change within these strains. And so this is a stacked volcano plot. And there we've taken a comparison between the lab-evolved strain and the wild type, so our resistant susceptible. We've also done a comparison between the clinical resistant strain and the susceptible wild type, and then plotted those on top of each other. And the proteins that are filled in are ones that were commonly identified. And we were particularly interested in 6 proteins that showed a significant increase in abundance in the resistant strains. And one of these was ClipX, which is an unfoldase protein. And it typically goes in a complex with ClipP, a protease that is associated with protein degradation. And so it helps to unfold these proteins that are tagged for degradation. And so we went to the literature and we looked for ClipX inhibitors, and our collaborator in Germany was working on a compound 334 and how it had inhibitory activity towards ClipX within a bacterial pathogen. So we've tested out the compound within the lab, and we saw that there was synergy between the inhibitor of ClipX and fluconazole to kill the fungal cells, even if they're resistant. And that gave us the first point that we wanted to then explore further within in vitro and in vivo models. So here we took our fluconazole resistant strains and we co-cultured with macrophage and then treated it either with fluconazole, the inhibitor, or the combination of both. And this allowed us then to see a significant reduction in fungal cell survival in the presence of these compounds. And we also did this within an in vivo model where we worked with the gene deletion strain for ClipX in the absence of fluconazole and the presence of fluconazole. And what was really exciting is that even though we see a reduction in virulence in the ClipX deletion strain, when we add fluconazole to the system, the mice overcome infection. They survived to the end of the trial, and we kept pushing out the date of the trial because they were healthy and doing well. And when we look at the fungal burden, it was significantly lower. So these mice are susceptible to fluconazole again when we do not have the ClipX present. And so that was a very exciting finding for us. And we wanted to look more at the mechanism that is behind this. And we looked at ergosterol content, which is the target of fluconazole. And within our resistant strains, this content is very high, whereas in the deletion strains, it returns more to the wild type level. And so this gave us insight into how resistance is occurring, but also how we can target it. And you'll see here then we can actually turn resistance on and off. So we can inhibit ClipX or delete it, and we can reverse fluconazole resistance. We can add in heme and iron and increase that ergosterol content, and it gets higher. So it's really quite an exciting system that we're working on, and proposes a new model for fluconazole resistance. Also demonstrates efficacy within the in vivo model to make fluconazole effective once again, and begins to give us some insight into the mechanistic regulation of that resistance profile. So proteomics has really allowed us and enabled us to tackle these 4 pillars against fungal disease, working with that pillar 1 to discover and characterize antivirulence compounds from natural sources. Pillar 2, investigating improved diagnostics through the organ atlas infectome mapping by time and space. Pillar 3, characterizing host-pathogen interactions for the discovery of new druggable targets and therapeutic intervention strategies. Pillar 4, then, looking at protein disruption and how we can reverse fluconazole resistance, making the drugs effective once again. And so with that, I'll end on announcing a couple of upcoming events that I'd like to showcase. We have the 6th International Plant Proteomics Organization Conference in May. So if you're a plant researcher, that could be something of interest to you. And the Human Proteome Organization World Congress, HUPO 2025, will be in Toronto in November this year. And then lastly, I want to highlight Moms in Proteomics. This is an international initiative that I founded in 2021. We have over 200 members around the world and provide travel awards, networking opportunities. It's just a really, really great group that we enjoy interacting, and everybody is welcome to join as well. And so I'd like to thank the members of my group that do the research, as well as our collaborators that support it, and our funders that enable us to do all of this work. And thank you for your attention.
Okay, Jennifer, thank you very much for that great talk. As usual, you gave a great coverage of your research area, and infectious disease and fungal disease is also always very fascinating. And so we're going to go ahead and do an unconventional Q&A session with you first before we open it up to the audience here, the attendees. We wanted to just dig into some of the technical aspects, but then also have a lot of fun with it and get to know you more as a researcher, but also the challenges you face on a day-to-day personal level. I know you've been very open with that in several of your initiatives and talks, and so we wanted to give you this extended opportunity. So I'm going to go ahead and jump into some technical questions, if you don't mind.
Sure.
So I'm interested in some of the unique challenges that you face with the fungal disease research that you've been doing. Just looking into how you compare that to bacterial and viral infections. I know a lot of people compare and contrast in these areas, but what would you say is unique about fungal disease research in of itself, in particular?
Absolutely. Thanks, Stephanie. One of the fascinating things about fungal infections and fungal pathogens is that everyone has a different baseline of exposure. So an individual may have, depending on the environment that they live in, the air they breathe, the food they eat, an infection can be very different for individuals. And your baseline, whether it be, your levels, change also throughout your life and through exposure to different things. And so that presents a unique challenge with fungal pathogens because we're looking to diagnose disease, but it's a hurdle to say if it's acute or chronic, latent infection, or perhaps it's an individual's elevated levels. So I think that's one of the unique properties about fungal pathogens and is compared to viral and bacterial. Identification is also a bit of a challenge too. We don't typically test for fungal pathogens in the clinic, and we don't always look for antifungal resistance. We have more mechanisms in place for bacterial testing. So all of those kind of present technical challenges that we try to overcome.
Okay. Well, I think you set up my next question pretty well, and that's what role does bioinformatics play in your proteomics research? Any tools that you're excited about that hopefully make your life a little easier? If you want to comment on that.
Yeah. So I would never consider myself a bioinformatician. I'm a proteomics researcher at heart, but it's amazing the ability of bioinformatics tools once you use them and the diverse applications. So of course we use them for our own proteomics data analysis. Whether it be, taking the raw data from the mass spectrometer and then looking to identify our proteins and then visualize our data. We've developed a new platform, ProteoPlotter, that combines with other platforms for ease of visualization and statistical analysis to just enable someone that is not a bioinformatician or perhaps not a computer scientist to get more out of their data. And I think too, then integrating proteomics and mass spec and data analysis with computational tools. There's so much machine learning and AI going on, and we have a lot of opportunity to integrate them, whether it be the technology development side and improving our detection methods or the data analysis side and really characterizing our targets so that we can do less in the lab and more on the computer to inform us what we should be doing.
Okay. So we are in this era of like big data, big data. You could never have too much data. But how do you integrate? I'm going to take this even larger here. How do you integrate various fields outside of proteomics to address some of the challenges that fungal pathogens present itself? Like, do you look at other, I guess, sources of information for your research?
Yeah, it's a great question. Like in the sense that in my talk, I go over One Health and how we look at the intersection of like animal health, human health, and the environment and how they all relate together. And so within that framework in my lab, we use different disciplines like microbiology, molecular biology, biochemistry, computational biology. All of those allow us to tackle fungal pathogens and fungal diseases from different ways, whether we're, trying to identify a new biomarker or characterize a new druggable target. But if we look more into drug discovery and we want to, start to move down that path, then we bring in more of the pharmaceutical side. And perhaps the biochemistry side gets a little bit more emphasis than some of the others. So I think part of it is integrating different disciplines. Another part is also learning from the different systems. So for fungal disease, we may study it in an agriculture context for some projects, or we may study it in a human medical context for some. But we can draw parallels in fungal virulence factors being produced, the immune response of a plant versus a host. And those parallels actually give us a lot of power to uncover new things that we wouldn't necessarily see if we were very single disciplinary in our approach.
Yeah, that's true. Okay, so I work for Thermo Fisher Scientific, and I have to do a personal plug here. Of course, I'm very passionate about our instrumentation, and I always like to come across collaborators and customers that are also very passionate about our instrumentation. So I'm going to ask you, maybe a bit of a thermo question. What future developments at in thermo mass spectrometry are you the most excited about, and how do you see thermo products in particular influencing your work?
hmm Certainly, the technology side comes to mind first. So, it's amazing developments with mass spectrometers over the past 20 years, but in particular, disruptive technologies such as the Astral in the past year and a half, 2 years. And that allows us to not only go faster and measure more samples, and again, if we're doing that in a clinical context, that brings a lot of power. But for us, it's also sensitivity. Typically, within a fungal disease context, the fungal proteins and peptides are much lower abundance than the host. And we've been previously limited in our ability to detect them, to monitor them, track them over time, assess their response during infection with previous approaches and technologies. And so now we're able to get better depth of coverage, and that's what we really want to push the envelope on. So that's what we're excited about, is can we identify even more fungal proteins? Can we find new virulence factors? Can we find new druggable targets that we never knew existed before because we couldn't even detect them? I think those are our key things. Of course, the computational side and combining the data with what we can actually do with it is also very important and very powerful. But the instrumentation is really what drives our innovation for sure.
Yeah, that's great. That's what I like working here for, all the innovation. So I'm going to stop on the technical questions because I want to leave some time for our listening audience to ask their technical questions. I'm going to jump into some maybe more personal and hopefully fun questions that at least we'll have fun with. So we're going to go ahead and switch to that. So what advice would you give young researchers, particularly women entering the field of proteomics, now that you've been doing this for a couple of years? Looking back, what advice do you have for them?
Yeah. Well, first of all, I found the proteomics community, the international proteomics community, and also the national here in Canada, to be, incredibly collaborative and also innovative and driven. And I think it's amazing to me that two people doing proteomics can be approaching it from very different perspectives and use very different approaches and tools and workflows and really collaborate and build something amazing. So I think one, it's to embrace that collaborative community and use that to help drive your own research forward. And the second one, especially for women in STEM, I think a lot of people tell us to learn to say no. And I think that's one of the biggest pieces of advice people will hand out is learn to say no. I'm very much like say yes to things. I find like if you say no to everything, you lose those opportunities or you don't know where they'll lead to. So for myself, it's like say yes, give it a try. It may not work. It may not be, worth your time in the end, or it may not be the passion that you thought it would be, and that's okay. You can always change later. But it's nice to give things a chance because you never know where they will take
I like that. I think I live by that too. You don't know until you try it, and you
you.
don't
Yep.
know
Yeah.
what you're going to stumble across. So that's great. Can you discuss the importance of mentorship in your career? How has that helped you, or your role as a mentor too? Because I know you spend a lot of effort on that part.
Hmm. I think mentors are, incredibly important at all different stages. And one thing that I heard once that I never really thought of is someone said, you have to have multiple mentors. And I always thought like I'd have one mentor and that was sufficient. But now when I look back on it, I think like, no, it's amazing to have mentors in all different areas. And so for myself, my PhD supervisor and how he led his group really provided a model for how I lead my own research group. And then I have others that are more, technology driven or that community and collaboration driven that I can go to and ask specific questions on. And they change too. I think another important piece is that being a mentor doesn't mean that you can't learn from your mentee. And there can be a lot of back and forth and a lot of growth in both from both sides, which gives importance to it, to the role as well. And so when I take on a mentorship role, I really try to tailor it to the needs and the goals of the individual, and also respecting that they may not need me as a mentor in a year, or they may come back to me in 10 years and say, what you said here is what I'm doing now. And I think having that flexibility and just being available is a really important piece.
That's great. So how do you stay motivated I can imagine research and having all these people that you're mentoring and activities that you have going on, conference involvement, all those things that you have going on. What keeps you motivated and inspired to keep going, keep up the research efforts?
Yeah. I would say it's certainly not getting back grant reviews or paper reviews. That's one that will squash motivation pretty quickly. But the drive to do something different, the drive to help a project move forward, or help a student get to their thesis and achieve their goals, that I find very rewarding. I also really thrive on the community-based activities. And so for conferences, you organize them, put a lot of work into it, but then you get to see all these people that you've worked with and you get to interact. And I think I find that really rewarding. And you get to see the difference that you're making, which is also a big driver for me.
That's great. Yeah, there's a big personal aspect to it as well.
Mm-hmm, mm-hmm.
Okay, so you founded Moms in Proteomics, and for those that are listening and haven't heard of this, I thought it'd be good for you to introduce it, but then also tell us, what was the best part about founding it, what your activities are like. If you want to go ahead and share that with everyone joining in, that'd be great.
Yes, absolutely. So I founded Moms in Proteomics in 2021, and it really was built out of the idea that we have these personal lives that we don't necessarily get to share or incorporate into our professional lives. I find as a woman in science, that's a big, I have a big wall there that I put up, and I think many people do. And that's not a bad thing. But I think with the COVID-19 pandemic, it really showed us that there's this huge intersection between personal and professional lives. And so when I first had the idea for Mums in Proteomics, I reached out to a few of my colleagues that are mothers in the field, and they really thought it was a great idea. And we hosted a panel discussion at the Human Proteome Organization meeting in 2021, and discussed, how do you balance work and life? How do you go about, having children in science? What sort of mentors did you have? What can you provide as a mentor? All different avenues. And now we've grown to over 210 members from 25 countries. And I'm able to provide travel awards through industry sponsorships, such as with Thermo, so that members of Moms in Proteomics can go to a scientific conference and present their work and showcase what they're doing, highlight their achievements. We've also partnered with Females in Mass Spectrometry to have different lunch and networking events and discussion groups. And we're working to build workshops as well as a commissioned book from the Royal Society of Chemistry, all towards recognizing and acknowledging and supporting mothers in STEM.
Okay, great. And then if those who want to get involved, how should they find more information on Moms in Proteomics?
Yeah, so momsinproteomics.ca, and there's a big join now button, and it comes right to me. The email comes right to me, and then I add you to my mailing list, and then you get our notices. So at scientific meetings and conferences, I tend to host networking events, so I'll send that information out. It's free to attend, and so you can come and join and meet other members. And I will emphasize that it is open to everyone. We have very many people that join. So you do not have to be a mother to join, and you don't have to be in proteomics either. We also have many disciplines represented too.
Yeah, I think it resonates with a lot of women in STEM, just talking about it with other fellow coworkers here at the office. But I still need to join too. So I gave myself my own action item, but I will. Okay, so how do you unwind after a long day of work, or all the activities that you have going? How do you unwind? What's your preference?
That's a good. I think if you ask my husband that question of how I unwind, She's always like very, very driven, I would say. But I definitely, for me, it's very important to have that balance. So I many times I will walk or run home from work, and that gives me a really nice debrief time to myself, and exercise. That's probably my favorite way to kind of unwind and let the day go, and start fresh. Because when I come home, it's dinner with the family, and then bedtime routine with the kids and everything. So it's kind of like preparing for the second marathon of the day, and that works really well. I also like to watch like trashy shows on Netflix and read books that are just like those fun summer beach reads, so that you don't have to think about everything else. It's just fun, and those are definitely highlights too.
It's great. Yeah, we all need something. Okay, so
Mhm.
one final question for me. If you weren't a scientist, I know that's a tough choice here, but what career would you have chosen if you weren't a scientist?
This is very easy for me to
Science
reply
is
to.
taken away from you. Oh my goodness.
Science is taken away. I would be a wide receiver in the National Football League. This would be my dream job.
Really?
I'm a big football fan, and I
Oh,
absolutely
great.
love athletics. And so you can combine like the two of them together, and that could be your career. That would be
Jennifer, we haven't talked about football yet. Anyways, I don't know what I would, I don't know how I would answer that question. It probably depends on the moment. But I think that is an interesting thing. We learned another thing about you. You like football as, yeah, it's an obsession lately. Or not lately, but in definitely is a popular sport to watch. Just had the Super Bowl.
amazing.
Okay, well, thank you very much for your time for those questions. But we're going to go ahead and hop over to any listening members that have listening people from the audience to go ahead and answer their questions. See what they have to say.
Fantastic. Thanks so much.
Jennifer, thank you so much for your informative presentation. And Stephanie, thanks for those great questions. We love getting to know Jennifer on a more personal level. Now, I'd love to jump into all of our attendee questions. But before we do that, I just want to remind our audience how to submit questions if you haven't already done so. Just type your question into the Ask a Question box. It's located on the bottom of your screen. And we will answer as many of your questions as we have time for. So we're going to squeeze a bunch in here. I also want to direct your attention to the far left of your screen where we do have some helpful downloadable resources. So Jennifer, I'm just going to dive in. Our first question here, can fungal infection trigger human body autoimmunity? Jennifer, are you able to hear me? I'm going to give Jennifer just a moment to refresh her browser. I think that she's not able to hear me at the moment. All right, bear with us 1 second. Okay. While we're waiting for Jennifer to get her audio connected, I do want to let our audience know that today's webinar will be available for on-demand viewing. And once this presentation is over, LabRoots will send you an email that will let you know when it's available for replay. And we do encourage you to share that email with your friends and your colleagues who may have missed today's live event. What looks like Jennifer's audio is not connecting correctly. What we're going to do is we will be answering all of these questions via the email address that you provided at your time of registration. So we're going to go ahead and answer these questions via email. I want to thank everybody for joining us today. And again, this presentation is available for on-demand viewing. You may also submit your questions during the on-demand period. So thank you all for joining us. We appreciate your time and your great questions. They will be answered via email. And to all our friends out there, thank you again for joining us. Have a great day, everyone. Bye-bye.